Today we're going to unravel the effects of an obesogenic diet on the brain. We will be discussing the results of two articles, both done in the NutriNeuro lab. While obesity is commonly discussed for its physical health implications, it also impacts cognitive function. Today, we're delving into memory alterations linked to adolescent obesity. Adolescence is a crucial period of brain development marked by intense plasticity. This means the brain is vulnerable to environmental changes during adolescence. In humans, studies have revealed the heightened vulnerability of the amygdala and hippocampus during this period. fMRI studies have shown that obese individuals tend to exhibit reduced volume in the hippocampus and increased activation in the amygdala. Studies have shown that the amygdala is crucial for emotional memory processing and that the hippocampus is involved in declarative memory. The articles we're discussing today stem from studies carried out in the NutriNeuro lab. In rodents, adolescence refers to the transitional period between weaning and adulthood. Boitard and colleagues have demonstrated that a high-fat diet from adolescence through adulthood induces spatial memory problems and reduced hippocampus neurogenesis. This was not the case when a high-fat diet was fed only through adulthood, showing the importance of brain development during adolescence. Later on, they discovered that exposure to a high-fat diet during adolescence also leads to increased synaptic plasticity of the basolateral amygdala, along with enhanced emotional memory. Now that we've seen the previous findings in the lab, we know that changes occur in the hippocampus and amygdala as a result of a high-fat diet, and that there's an association between memory disruptions and changes in the hippocampus and amygdala. Now, the articles aim to uncover whether synaptic transmission in these structures is impaired and whether they play a causal role in the deficits associated with a high-fat diet during adolescence. In order to study synaptic transmission, the authors established one control group that received a standard diet, while another group was exposed to a 2 to 3 month high-fat diet covering adolescence. In order to study the causal role of the hippocampus and amygdala on memory alterations, the authors used one control group and 3 high-fat diet groups, all subjected to a high-fat diet for 2 to 3 months. One group received only the high-fat diet, while the second and third groups were administered a virus containing dreads injected in either the basolateral amygdala or ventral hippocampus. Dreads are engineered receptors that are activated by CNO. When CNO is administered and binds to the inhibitory dread receptors expressed in neurons of the basolateral amygdala or ventral hippocampus, it activates the G-alpha-I protein. This activation of the protein inhibits adenylate cyclase, which eventually leads to a decrease in neuronal excitation. As a result, neuronal activity in these structures is suppressed. To investigate synaptic plasticity in these two structures, long-term potentiation was examined. This involved stimulating the entorhinal cortex and recording activity in the CA1 region and basolateral amygdala. For the study of a potential causal role, 2 behavioral tests were conducted, the object recognition memory test and the conditioned odor aversion test. The object recognition memory test evaluates the ability to remember familiar versus novel objects. Rats naturally prefer exploring new stimuli, so increased exploration time towards the novel object indicates good recognition memory. Conversely, a decrease in exploration time suggests impaired recognition memory, possibly indicating difficulty in remembering previously encountered ones. This type of memory is associated with declarative memory and relies on the hippocampus. The conditioned odor aversion test evaluates rodents' ability to associate an odor with an aversive stimulus. An increase in odorized water consumption after conditioning suggests a failure to form an associative memory between the odor and the aversive event, while a decrease indicates successful formation of aversive memory. This type of memory is dependent on the basolateral amygdala. The authors discovered that a high-fat diet lasting 2 to 3 months during adolescence heightened synaptic plasticity in the CA1 region of the hippocampus compared to the control group. This finding is particularly intriguing because a longer diet lasting 6 to 7 months reduces synaptic plasticity in the same region. Additionally, synaptic plasticity in the basolateral amygdala increased. The authors also found that this high-fat diet led to a decrease in recognition memory compared to the control group. Similar results were observed when inhibiting the basolateral amygdala. Interestingly, inhibiting the hippocampus restored recognition memory to nearly normal levels, similar to the control group. Regarding conditioned odor aversion, rats on the high-fat diet exhibited heightened odor aversion memory. This effect was also observed when inhibiting the hippocampus. Interestingly, inhibiting the basolateral amygdala restored aversion memory to nearly normal levels, comparable to the control group. To sum everything up, a high-fat diet lasting 2 to 3 months covering adolescence has significant effects on both the hippocampus and basolateral amygdala. Heightened synaptic plasticity is observed in both brain regions. Additionally, the authors demonstrated that alterations induced by the high-fat diet in the hippocampus contribute to impaired recognition memory, while alterations in the basolateral amygdala lead to enhanced aversion memory. This demonstrates that these structures play a causal role in the deficits associated with a high-fat diet during adolescence.